All right, well, as you've heard by now, I've decided that it's best to go ahead and record the rest of Lecture 8 so that we can stay on schedule with Lecture 9 and Lecture 10. And then I'm sure we'll be behind again by Lecture 11. Lecture 11 is going to be next Monday. And what I think will work well about this is that I'm going to go ahead and say I'm going to record an extra session after class next Monday. And then we'll get caught up with all the Lecture 11 stuff. We'll get caught up from this point. So that means that lectures 9 and 10 will be able to get out on time, and then I'll be able to add everything that we need for test 2 in a session after class on Monday. So if you can stay after class on Monday, you can go ahead, or if you need to put it into your schedule or something like that, go ahead and do that. But of course, I'll be recording it with the whole point of you don't need to stay if you don't want to, okay? But it really is probably the best way to learn and things like that. So let's go ahead and move along with where we were. Let's finish up Lecture 8. Then we'll be on schedule and we'll be moving right along. So we ended up with this, and we had this residues in alpha helix and beta conformation, and I said that you can get this from the angles from the Ramachandran plot. From the X-ray crystal structure that you get, you know where the XYZ of every single atom, or at least every single heavy atom, like carbons and nitrogens, and you can actually measure the angles between them. If they fall into the alpha well, you call it alpha helix conformations. If they fall into the beta well, we call it beta conformation. So this is one way to identify which residue goes with which. There's another way experimentally to actually measure which secondary structure you have, and it's called circular dichroism. I like this because I've used it myself, and it's shown me something useful in the lab. Circular dichroism actually is a really complex technique. If you notice that the y-axis is this delta epsilon thing, which has to do with ellipticity of the signal, it uses polarized UV light and the chirality of the carbons to be able to measure whether you have a curling backbone or whether you have a straight back and forth backbone or a randomly moving backbone, randomly twisted backbone, we should say. Those are the three options that circular dichroism can sort of distinguish between. And you have an example right here. If you put an alpha helix into circular dichroism, you get a signal like this, and it's actually in the UV wavelengths, as you can see. It doesn't really correspond to absorbance like you're used to because it's circularly polarized light and it's really weird. So it's best just to think of it as, if I have alpha helix, I get this signal. If I have beta conformation, I get this signal. And I get an actual negative signal, which is a signal that you can measure. You know, you can measure how negative it is. You get a measurable signal at about 220 nanometers. And in contrast, if you have the sequence just randomly coiling, then you actually get a no signal appreciably at all at 220 nanometers. So this is kind of cool. This is like where we had tryptophan and tyrosine and we could actually see them with UV light at 280. This allows us to see alpha helix and beta conformation and distinguish them from random coil. So if we have a signal at 220, then we say that we have the secondary structure is formed at that particular condition. And what this is really useful for is you can run a circular dichroism experiment at different temperatures. Basically, you just heat up the cuvette that you have the protein in, and then you can see what happens. If you look at this right here, this is data actually collected and used in one of my early papers, so I have a nostalgia for this. This is one of the first things that I published in my lab on my own. And what you see right here is I actually made the cooler temperatures cooler colors and then warmer temperatures warmer colors as you go up. And so here's the wild type, which means unmutated protein that we were looking at. And you can see you get a big dip in signal around 220. You heat it up to 37 degrees and you see the same signal that you saw at 25 degrees. But then you heat it up to 49 degrees and all of a sudden you lose most of your signal. You heat it up to 61 degrees and 70, 80, 90, you've lost a lot of your signal at that point. So you can see that this protein actually falls apart before half of it falls apart, at least the alpha helix part that you're looking at right here, will fall apart before you get to 49 degrees Celsius. But on the other hand, there is a mutant. This had 5 different mutations, lots of details that we don't need to know right now. But the important thing is look at the signal at 220. You have the same pattern as before, but instead of having lost most of your signal at 49 degrees, you've actually kept most of your signal at 49 degrees. This was an experiment that told us that this mutant protein on the right actually has more stability. It holds together more at higher temperatures, at least when you measure the alpha helix secondary structure kind of signal using circular dichroism. And this was useful, and by the way, this is not the strongest data, but I'm showing you because in conjunction with several other techniques, it actually showed us that the mutant appeared to be more stable than the wild type protein. And so that's actually kind of cool. This is actually a protein folding experiment, so it's a little bit ahead of the game, but it's, we'll talk more about these when we get to the protein folding part of this chapter. The important thing is it's showing you that you lose the secondary structure at different temperatures and that the protein on the right is more stable. So you can actually see alpha helix. If you heat up a sample, you can see the alpha helix signal go away. You can basically disrupt the hydrogen bonds and have the secondary structure go away. You can actually have one kind change into another kind, and that has a real-life application. So over time, you can actually have a protein aging, and it can sort of unfold from one structure and refold in another structure. And this actually happens in a way that you can see. If you look at a painting, you can see evidence that this has happened. So for hundreds of years, artists have known that they just found out by trying different things, washing egg white over their painting made the paintings preserve for a really long time. It's a really good preservative material. They didn't really know why, but it turns out that there's a protein reason why you should use egg whites as a wash to cover your painting and to keep it from oxygen damage. The reason is that over time, ovalbumin, we actually have it on this chart right here. I thought you did, but I guess you don't. Okay, but ovalbumin has mostly alpha helix structure, and the reason why over time the egg white will actually protect a painted surface, and you have right here an example of an egg white wash over the surface. Over time, just as it sits out, just at normal room temperature conditions, the egg white wash will change its secondary structure. You've still got all these alpha helices on it at first, and then over time the alpha helices will unfold and they'll convert into beta sheets, and so they'll kind of stretch out, you know, in a way. So the alpha helix content decreases by 73%, the beta sheet content increases by 44%, and they theorize that the beta sheet is actually more closely interlocked. I mean, it's literally a sheet hydrogen bonded to itself. And so this is a tighter fence. It's clear so you can still see the painting underneath, but it's a tight fence that keeps the oxygen from reaching the paint and fading the colors. This is why egg white is actually a good thing to use if you're painting a picture and you want it to last for centuries. So there are other ways we can talk about turning alpha helices into some kind of beta structure. Here's an example right here from a paper that studies hornet silk. It turns out the hornet silk has some really interesting molecular properties, and one of the ways that you can look into that is that you can actually pull apart the molecular properties. As you stretch it, it will actually change from an alpha helix conformation to a weird sort of beta sheet conformation. And so you have down here, you have a gel film of the hornet silk, and you see that that is not stretched very much. If you stretch it and move it over to the right, then you see that over here it's stretched, and when you look at the same film, you see a lot of what they call cross beta sheets. You stretched out the alpha helix into a beta sheet, not by weighting and not by normal room temperature unfolding and refolding like for the egg wash, but it's a similar kind of idea. You're just doing it by literally stretching the alpha helix out. Let me show you what that looks like. The important thing is that you actually have an arrangement of the alpha helices, where you have the alpha helices in a braid around each other. You have 3 alpha helices, and they're sort of braided together in what's called the coiled coil superstructure. This is an example of what they show you from the paper. You have the coiled coil on the left, and if you zoom in on that, you see that each of the 4 coils that they're showing here is an alpha helix. That is, so that's why it's a coiled coil. You have a coil of alpha helices, which are each themselves a coil. When you stretch it out, you actually will result in a beta sheet-like conformation. They're showing you here a parallel beta sheet just for conformation, but we just talked about what that looks like. The important thing is what you get after you stretch out the alpha helix is you get the structure on the right. You get a cross beta sheet that is sort of like a flattened alpha helix. But this really is a beta sheet, because if you look at the phi-psi angles, if you look at this, you have the little, 3 or 4 residues in a row before it does a turn and goes back, and you have actually a little anti-parallel, very short in this direction, but it's a true anti-parallel beta sheet. And I want you to imagine, if you can imagine how this happens, then you have a good idea of what these different secondary structures are actually doing. This is what they found with their paper. So the silk, when it starts out, is an amorphous sort of mix of coiled coils, which are alpha helices, and then some disordered parts. You see they're like little frayed bits of yarn there. And so if you have the alpha helices and then you stretch them, you go from the part C on the right, part C on the left, to the part 2, part C, in the middle, where you have the alpha helices sort of flatten out, and you end up with this cross beta sheet structure inside the coil. At the same time, you see what you've done on the top. If you look at the very top of the figure, you've taken all of these different jumbled sort of coiled coil domains, and you've pulled on them, so they've sort of unjumbled. They've sort of been pulled in a similar direction. But they aren't quite lined up yet. The important thing that they found is that actually converting the alpha helix to the cross beta sheet happens first. And it happens along with a general orientation, but the coiled coils are not completely oriented yet. That waits until more stretching. You have to put more force on it, and then finally all the coiled coils snap together. You get that in part 3 over on the right. Part 3 over on the right is where you have the coiled coils finally lining up. But the important thing is I want you to see how a little stretching in this direction can turn the part D structure for the 1D structure to the 2D structure on this particular figure. This is what they say. They say they apply tensile force under appropriate conditions. It induces a transition from coiled alpha helices to a cross beta sheet superstructure. And so the 4-stranded tertiary superstructure, and so notice that the coiled coil is one of our first examples. What does tertiary structure look like? Well, in the case of Hornet Silk, it looks like a coiled coil. The tertiary superstructure remains unchanged during the process. The coiled coils themselves remain the same, they just get oriented and they sort of snap together. But what's really changing when you're stretching is not the tertiary structure, but it's the secondary structure. And that was a surprising thing that they found. So you can change secondary structure just by stretching silk. You might have noticed on those cross beta sheets that they have to turn around really quickly. In fact, you have to have a structure. It's not just a meandering loop that you go. You have to basically have the amino acids twist as fast as they can to form the anti-parallel beta strands that they have there. So this actually is a defined secondary structure. It's sort of a third category. The 2 big categories are the alpha structure and the beta structure. And now here they have beta turns. There are different kinds of turns, but the important ones are the beta turns in this case. I don't believe it has anything to do with beta structure, except that you tend to see it associated with anti-parallel beta sheets. Because what it does is it's a U-turn that happens as quickly as it can. You have 4 residues in a row, and you have within those 4 residues, you basically do a 180 degree, you do a U-ey in the middle of the road with this particular sequence. Only a few amino acids are compatible with this particular at particular points. So in fact, if you look at this, there are actually two types of turns, but for example, they have slightly different things. If you notice that type 2, position 3 has a glycine. And that's not important for the specifics so much as it is that glycine is necessary to do the UE kind of turn like that. I don't really care that much if you know that it's at position 2 or position 3, but what I want is for it to make sense to you that glycine is required to have a very flexible, bendy turn like the beta turn requires. So the other thing that can be required, so what's required is that you have to form that H bond. Turns out that glycine can do phi-psi angles that others can't. It allows the type 2 turn to form that H bond. It's not a repeating H bond technically, it's a single H bond, but because it's defined by an H bond, we like to call that a secondary structure. It definitely qualifies as this level of structure, and it was on that little toy model that I brought out. Remember that they have the green U-turns. The green color was reserved for beta turn residues. They have characteristic phi-psi angles as well. So the other thing that is often at turns is you find prolene. The reason that prolene is in turns is, well, number one, it's already twisted up for its phi-psi angles in a particular way that turns out to work well with turns. But even more so, it actually can form cis omega bonds. Remember how we said that prolene is basically the only amino acid that can do this appreciably. And the cis bond, if you look at it, the trans bond sort of goes in a straight direction. The cis bond will actually go in a curved direction for this particular thing. And since you want the beta turn to curve sharply, the cis omega bond is actually what you want in this case. So that's why you'll find prolenes in these kind of turns as well. The last thing is you might think to yourself, okay, well, we have the angles that are good for secondary structure of these three types, the turns, alpha, and we have the beta sheets. But the other thing that you might notice is that maybe there's not any repeating structure at all. If you have that case, you have kind of the miscellaneous category, where you have, they can be any phi-psi angle that they can adopt, but it's just not a repeating phi-psi angle. It's not a repeating hydrogen bonding level that you can see. And so these aren't defined by having any secondary structure, so they are loops, is what we call them. Those are different than turns. Turns are UEs, loops are more gentle curves, and they can sort of meander back and forth. They are not the same thing as turns. Now, they are not necessarily highly flexible, but sometimes they can move. You don't know just because it's meandering that therefore it's moving from one angle to another. It could be meandering among fixed angles. And this is actually a question that's going to show up in the paper. They're going to look at it and they're going to say which parts of this structure are stable and really held together tightly, not moving, and which parts are moving. They are highly variable in sequence because, as you can see from this figure, they're usually on the ends of the molecule. And this is, they can usually, because it doesn't really matter. You don't have to form a particular structure with them a lot of times. So if there's not a part of them that's important for function, they're just kind of there. They're on the edge of the molecule. They're just sort of waving around. Like that one part that I showed in class. There was this part of the Sidera-Kalin model that I showed in class that was white. It was a loop conformation. And when we made the model of it, it was all wiggling around because it didn't have any bonds holding it down. So that's the one thing about it. If it doesn't have any bonds holding it down, if you mutate it, it doesn't matter. And so you see it mutate a lot. You can see gaps be put in. You can see different residues be put in. And this is one thing that makes loops stand out like a sore thumb when you're comparing different proteins. If you're comparing multiple proteins of the same family, then what you will often see where the loops are is you'll see all these gaps, you'll see all this variation. Remember that doesn't necessarily mean flexibility in space and time, but it does mean flexibility in protein sequence. And it probably means that this region of the protein is not important to protein function. So think about this, how would this show up in a sequence comparison? And that's where I have this for you. Where is a loop probably. Remember how I said this is variation, and it also doesn't fall under the category of the other two kinds of structure. Here's a sequence alignment for, it looks like 9 proteins put together. And the one thing that you can see about this is they have put particular beta and alpha regions above each region. And you see that those tend to be conserved. There's some parts of the sequence, however, that appear to be loops. They have to be in the middle of the protein, and they have to have a lot of variation in them. And in fact, since invariation, the invariant residues and the conserved residues are shown with colors in this place, the ones that are clear or white are the ones that are changing more. And so you can look for the big regions where there's big clear lack of color, lack of other secondary structure, and you can say the loops are probably there. So I'm going to give you a chance to look at your handout and see if you can circle where you think the loops are, or you can just stay here listening to me. Oh, and I thought I gave you the answer, but I'm not going to give you the answer because it's the way homework often is. I want you to be able to figure this stuff out for yourself. So I've given you enough information to be able to identify the loops, the interior loops of the protein, and I want you to look for them. Okay? There's more than one, I can say for sure. So this is what I already described, comparing alpha to beta propensity. This is the original data. This is when I converted it to be the same units as the other one. And this is where I normalized it to be where 0 is now the average value of the energies rather than simply being whatever alanine is. Because I want you to see which residues are more or less likely to form a particular structure based on the secondary sequence propensities. This is basically just based on their side chains. Now, I have this and I have the homework questions for you. It's important for you to see this is just one way to answer the question of what secondary structure will my sequence form. And remember that these are relatively small energies. It takes a lot of them in a pattern to add up. And that's why this way of looking at secondary structure through propensities can coexist with a compatible or complementary way of looking at how do I explain which sequence forms which secondary structure. Now, the reason why I'm going through all this is I have an experiment for you right here that goes through and it shows you that even if you have the exact same residues, how you arrange those same residues will result in different secondary structures based on their arrangement. Now, all of these residues are probably average in propensity. In fact, if we go back to this, we can probably say they use like histidines, and they might have used glutamates, and things that have sort of average propensities, or they have a good lower down propensity for each of them. Isoleucine and methionine are examples of ones that will have good propensity for either structure. If you have a good propensity for either structure, how does the protein choose which one it adopts? Well, the protein does what it does, and we're just trying to follow along and predict, see if we can understand why it does what it does. And so part of the reason why the protein folds the way it does must have something to do with patterns as well as propensities. In fact, this is probably more important than propensities because you can do what we're showing here. You can take the same amino acids and put them in a different order. And you can just put them in a polar-nonpolar pattern like this. In the polar-nonpolar pattern, if you do every 2 residues polar, every 2 nonpolar, if you think about that for an alpha helix, it would fulfill the rule that polar would be the same side of the alpha helix every 3 to 4 residues away from each other, and the nonpolar would form a single side of the alpha helix. So you'd have an alpha helix with a polar side and a nonpolar side. And that would actually help to fold up and to be a functional protein if you had enough of these helices next to each other. They showed that this will happen if you just make this pattern. And if you make this pattern for the A16 on top, you will get an alpha helical protein. On the other hand, if you take the exact same amino acids and you then take them and you do the pattern where you have every other amino acid, remember that that goes with beta sheets because every other amino acid on the beta sheet is either up or down. It goes up, down, rather than circle, 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 right? So because of this, then you end up with a beta sheet with a polar side where they're all happy with each other, and a non-polar side where they're all happy with each other. And those can be turned toward water or away from water into a beta sandwich protein. And so if you do this exact same residues, they end up being a beta sheet protein. And so that's the conclusion of this paper. Sequence periodicity, which means the pattern that is in the sequence, determines the secondary structure of this protein. So the other thing that you can do is, now that you know your amino acids, and if you know about these patterns, you can make a guess based on the patterns. Now realize this is not usually based on the propensities. I'm not expecting you to remember the propensities, except for the extreme ones like glycine and proline don't like either one. The extreme propensities you might be able to understand, might be able to remember, but the propensities really are kind of a subtle thing. You have to look at the numbers, and the protein has to add it up. But you can see patterns. And in fact, if you see patterns, you can tell that a certain sequence will actually form an alpha helix by just looking at it. That's what these researchers did in this paper. So I want you to look at this particular sequence. And they show with this sequence that you have like residues that are 3 to 4 residues apart. You have a pattern similar to what you had for the A16 protein on the last slide, except that this is a real-life amino acid, a real-life sequence that they found. And they're actually showing you in this figure, they put this figure in the paper to be able to say to their fellow scientists, hey, look at this. Isn't this cool? This is this is a alpha helix, because we have this pattern. So since they expect scientists to know that, they expect it to know it based on what I've just shown you. So now you can be expected to know it too. These patterns aren't absolute. Remember that proteins are not maximally stable. They're just stable enough to do the job. But sometimes, as in this case, they are strong enough to be visible to the human eye. So just remember, we want to summarize all this up by looking at the Ramachandran plot. And you can answer questions by saying that there are different things you can see in the Ramachandran plot. We've already talked about how you have the alpha region down here in the southwest quadrant and the beta region up here in the northwest quadrant. But there's a few other things that they found out. For example, parallel beta sheets are in a slightly different place from anti-parallel beta sheets. That's kind of interesting. There's certain phi-psi angles that are just slightly better for one rather than the other. The other thing that you'll see is that the right-handed twist ends up giving your beta structure phi-psi angles in this particular bean-shaped region they have here. But this can also help you understand other things. For example, we're going to talk about collagen in a couple of days. It forms a triple helix. And yes, it is a helix, but it's a helix in the same way that that stretched out wasp hornet protein is a helix. It's a helix because it's 3 helices of strands around each other. Just because it's a helix does not mean it's an alpha helix. Sometimes these helices can be stretched out helices, and the collagen triple helix is a good example of this. If you look at the phi-psi angles for the collagen triple helix, you'll see that they don't fall in the southwest quadrant, they fall in the northwest quadrant. So that means that the collagen triple helix is actually more like a beta structure than it is like an alpha structure. The other thing that you can see is that you can have left-handed alpha helices. It's a little bit harder to do that for the amino acids because of the chirality of the amino acid, but it's definitely possible to have a left-handed alpha helix. It's just not as common, and you only see it, honestly, for short stretches when you're looking at proteins. And this gets back to the question that we had in class today. One of the reasons why I wanted to go ahead and cover this today. This is what happens when you take rabbit pyruvate kinase, just a protein crystal structure, and you take every amino acid and you plot it out on a phi-psi plot. You get kind of what you expect. You can see that you get a lot of residues that tend to fit within the wells. And if they're outside the wells, they're not outside the wells very far. In fact, the residues that are outside the wells are more likely to be computational mistakes than they are to be truly twisted residues, twisted in that way. It just takes a lot of energy to twist the residues in that way. And it's more likely that you made a mistake on your computer or you don't have the resolution to really get the protein to where it's supposed to be. That said, you can look at this and you can ask questions like, okay, is this protein likely to have both alpha structure and beta structure in it? It looks like I see a lot of evidence for each kind of structure, but which kind of structure does it have more of? It looks like the alpha structure is a little more dense. You have more amino acids, more densely packed there. It looks like more dots overall. So I would guess that this is about 2/3 alpha structure and only 1/3 beta structure for the types of structure that we can see in this way. So that's one thing that you can do. You can judge the quality of a structure, and you can also see how much beta versus alpha it has in it. Last thing I just want to mention, don't forget about our friends the disulfide bonds. They are covalent bonds, but they're weak covalent bonds, and so you can break them pretty easily, and proteins and cells can break them pretty easily too. You have enzymes that actually will go around and sometimes break and reshuffle these bonds. And so are they secondary structure? Are they tertiary structure? Well, they're kind of their own thing. And so disulfide bonds tie the protein chain together, but they are permanent structures that you can even break all the hydrogen bonds. Covalent bonds are stronger than hydrogen bonds, and so you can break all the hydrogen bonds, break all the secondary structure, and you still have the disulfide bonds there. Just remember that DTT, which we mentioned earlier, is one way to break them. And also beta-mercapta-ethanol is another one. I mentioned this because it shows up in the homework somewhere, okay? But both of these are thiol molecules that will reduce and break the cysteine-cysteine sulfur-sulfur bonds. But it's important to know where those cysteines are, because that's part of understanding the structure of the protein. That brings us up to date. And so please do go ahead and definitely get ready for the reading quiz, get started on the homework, get started reading, and I will see you in lab this week or in lecture. All right.